So good afternoon, everybody. Welcome back. I'm going to continue on the theme that Tony very eloquently introduced of post-translational modification. And so this is really my version of one of Tony's opening slides, which shows phosphorylation and dephosphorylation. But it could just as equally represent ubiquitination and deubiquitination, or summylation and desummylation, acetylation, deacetylation. It's this kind of reversible process that the cell uses to tag proteins to transfer information. Now, the sort of wrinkle to the story that I'm going to bring is simply that we believe that this happens in the 3 dimensions of space and the 4th dimension of time. So space and time are key elements. If you don't bring things together at the right time, nothing can happen. And the model that I will use to sort of introduce this concept really comes from trying to grapple with the complexities and the pleiotropism that's built into cyclic AMP signaling. And one explanation for that is that you have a family of anchoring proteins that sequester enzymes close to their substrates at precise locations within cells. And it's really from that this family of molecules that we've been sort of instrumental in discovering and working on over the years, the A kinase anchoring proteins came to being. Because as their name would suggest, the anchor protein kinase A. But clearly, for them to be more general signaling effectors, they have to be involved in bringing together many more combinations of signaling molecules, such as other protein kinases, and importantly, very importantly, signal termination enzymes such as phosphodiesterase, phosphatases, and phospholipases. So through these modular interactions, and we've already evoked the beautiful work of Tony Pawson, but through these modular interactions, you can create quite sophisticated macromolecular assemblies that can account for the integration, the processing, and the propagation of cellular signals at unique locations within a cell. Since we began this work, there are now at least 47 different human genes that encode ACAPs. There are multiple splice variants, and as a result, work from many, many labs has resulted in what is called here the ACAP landscape that gives you a 3-dimensional sense of how proteins are organized and how they're clustered together in different combinatorial assemblies. And this is another way to take advantage of the limited number of gene products within the genome, add on top of that covalent modification, and you can begin to reach the complexity that's necessary for the intricacies of human life. So this is spatial organization. But as I mentioned, time is equally important. And to understand time in the context of these molecules and these complexes, I'd just really like to focus on these 2 anchoring proteins that we've identified that are physically associated with ion channels. Because that gives us an opportunity, by combining fluorescent imaging, knockout technology, which is very appropriate to be talking at a Smithies Symposium about, and electrophysiology, to look at different elements of a signaling complex where you've artificially produced a complex where you've rescued an ACAP fused to a kinase activity reporter, in this case it's targeting protein kinase C, to an ion channel which the kinase regulates. So by taking these 2 different parameters, changes in the conductance and the activation of this kinase activity reporter, which is the warmer colors in FRET, you can begin to coordinate 2 events in the signaling pathway. In this case, this is muscarinic suppression of the channel, and that requires phosphorylation and activation of protein kinase C. So when you look at the temporal synchronization, when you remove the anchoring protein, these 2 events are uncoupled. They're uncoupled by about 11 seconds. It's not very long. You bring back the anchoring protein, and these 2 events become almost instantaneous. If anything, the phosphorylation activation happens ever so slightly before the channel effect, which is what you would expect. So an ACAP affords you, in this particular context, an 11-second advantage. Why is that important? Well, if I were to tell you that this channel is a pacemaker channel that controls the beating of your heart, which happens approximately once every second, you can understand why you would not want to uncouple this system. You would not be laughing at this point. And added to that, there are now mutations in some of the ACAPs coupled to these potassium channels that affect the signaling that result in arrhythmias. So there's a fairly good biochemical, cellular, and genetic basis for organization spatially and temporally through these anchoring proteins. So with that introduction, what we've done in the last few years is really moved away from trying to define these complexes, which I would describe as a sort of molecular stamp collecting, towards understanding how they perform local signaling mechanisms in the context of disease. And the diseases that we've focused on, and this might be in relation to what's happening to me, are age-related and chronic disorders. What do I mean by that? Well, specifically, changes in glucose homeostasis that underlie diabetes, hypertension and cardiac hypertrophy, one of the major symptoms of diabetes, and visual problems such as cataract and retinopathies. And it's really the cataract that I'm going to talk about today. Now, the way that we study these traditionally has been to start with the system, whether it be a mouse or a human, move towards the cell, and then drill down towards the molecular organization and the molecules. And it's that roadmap that I'm going to follow today in the context of this complaint of cataract. Now, cataract is induced by diabetes, but it's an extremely common problem as people age. In fact, it's the decline in ocular transparency that afflicts 90% of individuals by the age of 70. What is it? Well, it's a pathophysiological consequence of defective water and nutrient circulation in the lens. If you look at me at the moment, what you're trying to do is process the light in as clear a manner as you can through your lens so it hits the retina, sends signals to your brain, and you realize that somebody's talking to you. And you can see that, even though I'm dressed in black, you can still see it. For that to happen, the light has to go through the core cells of the lens. Those cells are highly differentiated to have as few organelles as possible, because you would get diffraction and things would get fuzzy. Added to that, you don't want a conventional vascular system to deliver nutrients through the blood, because if you have arteries and veins there, you're going to be seeing red, which obviously in Wisconsin might be a good thing, but coming from the other university, UW. But it's not going to be particularly advantageous from an evolutionary point of view. So therefore, nutrients and small molecules transfer through these cells through gap junctions and through these structures called the major intrinsic polypeptides, or the aquaporin zero water channels, that can compose about 80% of the plasma membrane proteins in these core fiber cells. And this is a beautiful structure of this 6-transmembrane protein with this regulatory domain that was solved by cryo-electron crystallography by my friend and colleague Tamir Gonen. And that's really where the story begins, because through many conversations with Tamir, we discussed this paper I discovered where persistent pharmacological inhibition of protein kinase A promotes the onset of cortical cataract. So inhibition of a phosphorylation event has pathophysiological consequences. We reasoned that there would be an ACAP involved, and to cut a very long story short, because this has been published, we identified the anchoring protein. It exists in this particular context, bound to an 8 amino acid segment in the C-terminal tail of aquaporin zero, to position protein kinase A to phosphorylate this residue, serine 256, which, as Tony mentioned at the end of his talk, is in an unusual context for a phosphorylation event because it's within a helix, but it's within a regulatory domain. Here's the phosphorylation. This is a a phosphorylation screen of mouse lenses to show that this is the principal, if not the only phosphorylation site in this particular water channel. Here's the mass spec data, and it's absolutely conserved across species. So it's an important phosphorylation event, conserved primary structure, most often than not represents conserved function. Even though it's an important phosphorylation site, you can induce its phosphorylation. This is shown by quantitative phosphomass spectrometry using phosphoproteomics. We can induce phosphorylation almost fivefold, and we can block it by treatment of the extracts with PKI. So the take-home message is that this ACAP, which is called ACAP2, name doesn't matter, positions pKa to efficiently phosphorylate serine-235 of aquaporin 0. Why, and what's the mechanism? So unfortunately, the end of this is cut off. I don't think it'll affect my talk too much because this is just a side view of the water channel. This is what it looks like if you're looking up from one cell to the next. It's a tetramer, like many ion channels. The regulatory domains are at the C-terminal, are at the periphery of the channel, and the active pore are shown here. So this is where the water would go through here, 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 here. This is in the active phosphorylated context. What's been shown, if you dephosphorylate this channel, you allow the binding of calmodulin, which is shown in purple here, that actually cinches together two of these helical regulatory regions to close the water pore. And this is just some electrophysiological data that supports that concept. So the issue that really comes to play is what is happening to that phosphorylation site at serine-235. So here we have the structure of this helical domain in the non-phosphorylated and the fully phosphorylated form. Here's the two peptides. If you use circular dichroism, they remain predominantly helical. There's slightly less helicity in one form than the other. But the important point is that the negative charge that's provided by the phosphate and the bulk sterically prevents the binding of calmodulin, because this region would have to fit into this negatively charged calmodulin binding cleft to shut the channel down. So that's what we think phosphorylation could do. It's to prevent calmodulin binding, which closes the channel. We'd reason to believe this empirically by just doing binding experiments where we're looking in vitro at calmodulin binding to the apopeptide or the fully phosphorylated peptide. But I think more compelling evidence really came from genetics, when we were able to screen cohorts of cataractogenic mutations. This is just one case, which rather surprisingly, you get a very, very conservative mutation of an arginine to a lysine in the context of the phosphorylation site. Now, under most conditions, I would think that that would actually make this a better PK phosphorylation site. But if you look at the data, this site cannot be phosphorylated. And these patients are born with cataracts, and it's an autosomal dominant effect. So what we think is happening is that somehow these mutations that prevent phosphorylation of this site, there's also a cysteine mutation at this position that's been identified now, constrain the helicity of the peptide to prevent the unfolding for phosphorylation. So phosphorylation is an absolute prerequisite. So phosphorylation of serine-235 blocks calmodulin binding to enhance water conductance. And you need that flow in your eye to keep everything moving from one cell to the other to prevent pathophysiological damage. So this is all the sort of system set up to show you this one sort of hypothesis-driven experiment that allowed us to test this hypothesis. And this was to either disrupt this protein-protein interaction or disrupt this protein-protein interaction. The consequences of that would be to diminish phosphorylation, because you can get dephosphorylation by an associated phosphatase. Loss of phosphorylation promotes calmodulin binding. Calmodulin binding closes the water pores. There's a preponderance of the water pores at the two poles of the cell, and as a result, the movement of fluid is perturbed, and you go from this normal pattern of staining here to this rather ugly, blebbed set of cells that result from changes in water movement and osmolarity that, if you zoom out, gives you what in fact is in essence a model for the onset of cortical cataract. Now the difference is that this can be induced by these peptides, these anchoring disruptor peptides, within 72 hours. Cataract in most humans takes somewhere between 55 to 72 years. But nonetheless, we have a good model for how this works, and it really allowed us to conclude that anchored PK phosphorylation of aquaporin 0 is a homeostatic mechanism that sustains lens clarity. And this is just measuring the lens clarity, and you can see here, using standard techniques that opticians use, you've got this cloudiness that's emblematic of cortical cataract. And we believe that you need to maintain this phosphorylation site. So this sort of potentially clinically relevant finding leads to a very basic question about phosphorylation. Not just the mechanism of action of phosphorylation, but it becomes an issue of how does this happen. Here's our channel again. Here are our phosphorylation sites. Now we know that we need to have complete phosphorylation maintained of these residues to optimally keep the channel open. The question becomes, since each one of these yellow regions can also bind an A cap, and the A cap can bind 4 molecules of the pKa holoenzyme, do you really need up to 8 moles of catalytic subunit of pKa to ensure phosphorylation of 4 residues? Now, either you do need that because it's a solid state environment, or intrinsic disorder and flexibility built within the pKa holoenzyme could account for the movement. So that goes from a biological and a pathophysiological problem to a very clear structural biochemical problem. So we decided to solve the structure of the pKa holoenzyme to an anchoring protein that binds a close relative of ACAP2, is ACAP18, and the channel in this case is in the kidney, is the aquaporin 2 water channel that works through a very similar mechanism. And we decided to solve the structure not by conventional X-ray crystallography, because that had been tried for almost 20 years, in part successfully, and I'll show you some of the work from Susan Taylor that's done that in a few minutes. But we used cryo-electron microscopy and negative stained single molecule EM to be able to look at the conformation of the intact complex. So here's the 5 proteins in complex. Here's the negative stain EM. You have to have very good eyesight to see anything at the moment. I've highlighted these. They look like 3 beads on a chain. When you collect enough particles, and enough particles is somewhere in the order of 11 to 12,000 particles, and take advantage of a software that uses state-of-the-art face recognition software details to put the class averages together, you begin to get something that makes a bit more sense. So here's some class averages. Each of these has in the order of 800 to 600 individual particles sandwiched on top of each other. So here's one perspective, here's another perspective, and here's another perspective again. It gives you confidence, but it doesn't really tell you where everything is. Using immunogold labeling, we could identify that the anchoring protein was actually this part here. And then by taking advantage of crystal structures that we had done ourselves for fragments in collaboration with David Barford, and a lot of elegant work that's been done by Susan Taylor at UCSD over the years, we could begin to put together the molecular jigsaw that brought this complex to life. So in green here, we have the regulatory subunit, which is a dimer. In space-filling models here, we have the docking dimerization domain, which is the part that binds the ACAP. The ACAP fits very snugly in here. These are the cyclic AMP binding sites and the pKa catalytic subunit binding site. And finally, we could begin to see an anchored pKa holoenzyme. And immediately on looking at this, there were several things that became immediately apparent to us. First of all, the beads on the chain analogy is a very good one because these regions of structure are separated by about 30 amino acids that are intrinsically disordered. That explains why crystal structures have been really difficult for this entire complex. The other thing is that you could see the modular structure of this complex. You have an anchoring domain where you have protein-protein interactions that could be used to tether the complex at some point, with flexible linkers attached to two transduction domains where cyclic AMP can activate pKa in these parts of the dumbbell. So this was a combination of structure, of order, and disorder. And I think that this is also where EM analysis has some advantages, because you can look at your class averages where you have two-dimensional representations of many, many different configurations of your complex. And taking that to heart, here's a representation of some of these particles where they're organized in the order where the two transduction domains are closest together till they're furthest apart. There's a Gaussian distribution, but when you solve the structures of these in 3 dimensions, you can calculate that in the most constricted conformation, there's somewhere in the order of 250 to 300 angstroms between these two regions. And in the extended structure, it's approaching 400 angstroms. So you've almost got 200 angstroms of potential movement through these flexible regions of about 30 amino acids. One of the other things you can do is take your class averages and make a montage which gives you the boundaries of how a single complex might move. And when you see this and you can overlay on top of this the actual higher resolution structures, it gives you some idea of what might be happening. Certainly in the context of the aquaporins, a single anchored complex has enough movement to phosphorylate 4 sites. Alternatively, and it's always bothered me, why is pKa a dimer? Well, maybe it's because you have an anchored static site that then can service 2 independent substrates with other secondary protein-protein interactions to give you 2 different phosphorylation events for the price of 1. So that certainly gave us the first part to explain the question with aquaporin, which is how we sort of began this, but it really opened the door to many, many more fundamentally important questions about spatial organization of kinase activity. So certainly we favor this model. You could have 2 molecules in the complex, but I think 1 is going to be enough. But then it gets down to the issue of how far do these complexes actually move, and how far does the C subunit, the active part, the kinase end, have to go to remain active. So one of the ways to do this was to look at this complex in living cells. And in order to do this, I'll introduce a technique that I'm going to use quite a lot of in the last part of the talk, and that's fluorescence resonance energy transfer. Thanks to some beautiful work by Roger Chen and colleagues over the last decade, these kinase activity reporters have been developed where FRET-based activity can be used to induce activation by bringing a complex together to give you a FRET signal. And this is stimulation of cells with a physiological agonist isopaternal. You can see the warmer colors that give you the spatial and temporal pattern of pKa activation. But in the context of an ACAP, because this is a reporter that we made, which we called ACAR 18 RBS, where everything's localized. Now, you can see a fairly robust signal. It means nothing unless I think you can do the following control, which is you mutate that phosphorylation site and you abolish the FRET signal. And that's shown here. So this is a control experiment that just validates the system so that this sort of signal to noise ratio you have here, which is relatively subtle, is physiologically relevant. So this is with isopaternal. What we did next was to take advantage of this and think about what would happen if we changed the length of the linker that was holding the anchoring domains and the transduction domains together. We could use the wild type system with the reporter. We could make a compact form where we actually removed the flexible region, or we replaced it by creating a chimera using the zebrafish sequence. Remember, zebrafish are cold-blooded animals. They live in the tropics, but still, they're fairly cold-blooded. It's 53 amino acids, so it's 23 amino acids longer. So we had these 3 constructs, and we repeated those experiments. And what we discovered, kind of to our surprise actually, the shorter the linker, the more efficient the phosphorylation. So this is the primary data. I think really the amalgamated data tells a better story because it gives you the dynamics. The green line shows the truncated form. The extended and the wild type are pretty similar. There's no statistical difference. So the take-home message from this was sort of surprising. Although you've got intrinsic disorder, proximity enhances catalysis. So how close you are to substrate is going to govern how well it's phosphorylated. It's probably also going to make sure that you have less dephosphorylation or less effective signal termination enzymes. So that was the next thing that we wanted to look at. And that was to look at a substrate within a complex where we knew in a very defined way the distance of the substrate from the anchor kinase. And that distance, which is shown here based on our EM calculations, was in the order of 100 to 150 angstroms. It's not very far. And this is where, rather than adding cyclic AMP, we were looking at cyclic AMP independent phosphorylation. So proximity is what's really driving this. The data is shown here. We looked at phosphorylation in the presence or the absence of the anchoring protein. We were making these these complexes in vitro, so we had high control over what we got. And there's about a 2-fold increase. We can make that a 4-fold increase if we delete the linker region. But the take-home message from this is that we believe that we have a solid-state signaling event where proximity augments the basal phosphorylation of substrates. So with this piece of information, it wasn't really a hard conceptual leap to consider the possibility that even though you have cyclic AMP produced, and cyclic AMP is an important second messenger, it might not completely dissociate the PK holoenzyme complex. And as a result, the ACAPs control spatially and to some extent temporally all phosphorylation events. So the first sort of hint at this was again a biochemical experiment. We reformed our holoenzyme complex, but this time in a 50,000-fold molar excess of the second messenger cyclic AMP. Now yes, you can see there's less catalytic subunit here, but by no means has it all disappeared. So the C subunit remained attached, or some of it remained attached to the complex in the presence of high concentrations of cyclic AMP in vitro. We then did analogous experiments inside cells. These are 293 cells expressing ACAP18, where we could isolate the complex with an antibody to the anchoring protein. And using a physiological concentration of isopaternol, we could show under these conditions that we were able to recover the same amount of catalytic subunit irrespective of whether we activated the system with isopaternol. Now you could argue that this is a transfection effect or isopaternol wasn't working. Here's the loading controls for the lysates, and here's a phospho-PK antibody showing the cyclic AMP signaling pathway is turned on under these conditions. So this then sort of brings us back to drugs, because the most clinically effective drugs that work on the cyclic AMP signaling pathway that I know are phosphodiesterase inhibitor drugs. They're used in some cases or have been tried to be used as antiarrhythmic drugs. They're used as antidepressants, and they've been used to control asthma. Rolapram targets the type 4 phosphodiesterase, of which there are 73 isoforms, and milrinone targets the type 3 phosphodiesterase. As you can see, treatment with Rolapram abolishes and allows release of cyclic AMP from the complex, whereas milrinone has no effect. Well, it turns out that the binding protein that we were looking at as the substrate before was the type 4 phosphodiesterase PDE4D3 that would be affected in this system. So there's some selectivity within that pool of complex. We did have similar results using different anchoring proteins, but the take-home message here is that cyclic AMP stimulation with a physiological agonist promotes minimal dissociation of anchored pKa. So isopaternol is used to stimulate your heart, and that's right within the physiological concentration range. So that made us think about looking back into the literature. I'll tell you one more thing before I do that. We now wanted to use rolopram as a drug, as a probe inside cells to look at these protein-protein interactions. And in order to do that, we actually had to develop a different FRET-based sensor. So this is a sensor that we've adapted. There was a version of this originally developed by Manuela Zaccalo. We've modified this with different reporters to improve its efficiency, where we're looking at dissociation of the yellow and the blue fluorescent proteins from the anchored complex in the presence, in this case, of a physiological concentration of isopaternol. If you look here, you're not going to see very much of an effect because, in fact, there's very minimal dissociation under these conditions. And you can see here that the cells, there's 2 cells there, very little changes. More importantly, when we do these experiments enough times, we see a very minimal decrease in the signal. In fact, we think that there may be little or no decrease, and I'll show you some more of that later. However, when you treat with rolopram, which targets the associated phosphatase, you can get dissociation of the complex. So you're creating supraphysiological concentrations of the second messenger to get dissociation, while the normal physiological concentrations seem to be enough for activation and for phosphorylation of substrates. So that really leads us to this idea that minimal release of pKa happens under physiological conditions. Here's the controls. First of all, did we stimulate cyclic AMP? So this is a cyclic AMP sensor that shows with that level of isopaternol, you're turning on cyclic AMP. And also, again, using isopaternol under these conditions, we're getting activation of pKa, even though when we use the FRET sensor that looks at the protein-protein interactions, it's remaining as an intact complex. So these are under normal physiological conditions. The complex seems to be staying together. What about physiological conditions? Many studies, in fact, the last time I looked, in the order of almost 10,000 papers have been published using this diprene from the coleus, the Indian coleus plant forskolin, as a potent chemical activator of adenylyl cyclase, often in conjunction with a general phosphodiesterase inhibitor, IBMX, as a means to activate pKa. It's an artificial means to activate pKa. When we use this, we could show dissociation of the complex biochemically and using our FRET system. But what was very noticeable when we did those studies, this is on the same scale as I showed you before. You have this massive induction of cyclic AMP. And this is actually, in this case, in the absence of IBMX and an activation of the complex. So many studies that have looked at dissociation of the anchored pK complex are using supraphysiological conditions. They're using artificial conditions, which was somewhat surprising to us. So just to sort of conclude with this, we've gone back to another agonist, which I'll show you in a moment. This is an agonist, prostaglandin E1, which is a very well-used physiological agonist, and show you basically the same data as I showed you before, where under these conditions with a second physiological agonist, we see exactly the same effects in terms of release of the catalytic subunit from the anchor complex. It doesn't appear to happen. Here are the controls, again showing that you've got cyclic AMP synthesis and pKa activity. So this suggests that these macromolecular complexes remain intact and solid state signaling can occur in a cyclic AMP dependent manner. And so the sort of final set of experiments that really tested that a bit further was to see whether we could dissociate this complex as we predicted we could with rolopram, but then remove the agonist even in the presence of rolopram and see if we could reform the complex. And that appears to happen inside cells. So it's a very dynamic process. So let me sort of illustrate this more graphically just with this data as it's presented here. And you can see here that under the conditions where you've got agonist plus rolopram, you get full dissociation of the complex. But very rapidly, when you remove the agonist but leave the drug behind, you can get reformation of the complex. So let me put this all together in a more sort of diagrammatic form where I bring into play the structure. So here is the structure of the ACAP18 pKa complex at its extended form. There's somewhere in the order of 400 angstrom range of action. There may be some release of the catalytic subunit, but a very significant proportion of it stays attached. Because of the intrinsic disorder within the molecule, that can move down to a range of about 150 angstroms. And then given that this also binds a phosphodiesterase, that range of action is probably truncated further to an as yet indeterminate number. But it means that the area and sphere of activity for cyclic AMP is considerably smaller than we thought. We think that this happens within a range of about 400 angstroms. So here is a sort of pictorial view of some of the ACAPs that we've worked on. And the important point is that in each case, you're going to have some semblance of this configuration. So the idea would be that each of these ACAPs has approximately a 400 angstrom range of action. Now, if you sort of think of this in a cellular context, and this is actually taken from a Scientific American article that Tony Poston and I wrote several years ago, the idea is that each of these green areas, which could represent an ACAP complex that has this sort of range of motion, means that within a sphere of about 150 to 400 angstroms, you could get very efficient phosphorylation. So this somewhat radically changes the way that we think about local signaling for cyclic AMP. It certainly brings into play the question of how would cyclic AMP be either maintained or channeled to these positions. There are soluble adenylyl cyclases as well as membrane-associated adenylyl cyclases. But clearly, the spatial temporal organizers that seem to be playing a prominent role here are these anchoring proteins that position these molecules at these particular sites. So with that, I'll conclude and just really summarize what I was talking about. In a physiological context, anchored pKa phosphorylation maintains lens transparency. From a structural point of view, unstructural regions of R2 regulatory subunit influence anchored pKa action to allow this sort of reach round. And tethering within about 100 angstrom targets to favor cyclic AMP independent phosphorylation. We call this as solid state signaling. And physiological agonists seem to produce minimal, possibly no, dissociation of the anchored pKa complex. So it's a much more local and organized system than we initially thought. And these ACAPs are taking on a more prominent role to not only organize cell signaling, but also to segregate and insulate cell signaling so you can have multiple things happening in the cell at the same time. And that may be why you need so many protein-protein interactions that increases this sort of contextual selectivity within the cell. So with that, I'll stop and give credit where credit is due. The vast majority of this work was done in my lab at the University of Washington by Donaldson Smith and by Jessica Esseltine. We got a lot of help from our friends and colleagues in various parts of the world, particularly Steve Rakow and Tamir Gonen at HHMI Genelia Farms, and David Barford, who is now at the LMB Cambridge, that's helped us with some of the structural work. Thank you very much.
Very interesting, thanks.
Thank you.
I have two questions. The first question, in healthy lens, so the aquaporins has to be kept constantly open. That means the phosphorylation has to be persistent, constant. In that case, it will be fairly energy consuming unless ACAP can keep phosphatase away. The second question is this anchoring protein itself anchored to membrane, for example, through myristoylation or prenylation?
So let me answer the first question first. It's a very insightful question. It's a little bit more complicated than that because there are ATPases, there's adenylyl cyclases that produce cyclic AMP. There's also waves of cyclic AMP that go through the lens, and there are waves of calcium that go through the lens. And there is a phosphatase. So what that is thought to do is actually create a sort of peristaltic motion, because the other way to deal with this would be just, since it's a negative charge, why don't you just mutate the serine to glutamic acid or aspartic acid? But this peristaltic motion helps the processing of water movement, because you've got these polar to equatorial sort of eddies of flow of water from one cell to the other. So the peristaltic motion is produced by the sort of cyclic production and utilization of two different second messengers. It's very complicated, but that's thought to increase the movement of water from one cell to the other. And there's also evidence that cyclic AMP can move through gap junctions from one cell to the other, which is only going to increase that process. For the second part, I'm often asked how ACAPs are targeted to various different intracellular organelles or membranes. Several of them are myristoylated. In fact, the ACAP that I'm talking about, one of its isoforms is palmitoylated and myristoylated. Some of them are myristoylated and palmitoylated in the middle of the protein. Some of them have mitochondrial targeting sequences, ER targeting sequences. So there's a variety of ways that they can be targeted. Some of them have polybasic regions that allow them to attach to the interface of membranes. And some of them can bind integral membrane proteins to target them as well. So there's a plethora of targeting mechanisms.
That was really fascinating. I have two questions, and they may be related. One is to ask you if you might discuss what fails with aging, if that's known. And the other is to ask about the neuromyelitis optica connection, if there is one, where autoimmune responses to the aquaporin channels are in some way related to the visual problems. I wondered if the antibodies detect one or the other form of the channel. Is this a response to some abnormality, or is it the immune system causing some kind of pathology?
So there's several parts to both of these questions. So let me at least start with the first part, which is the issue of why does it take 55 to 70 years to develop cataract, yet we can do it in 72 hours. We don't really know the answer to that fully, but there are other factors that come into this, and we think it's the control of the phosphatase. And there's two ways to do this. One thing would be to either inactivate the phosphatase, or another idea that we've come up with relatively recently, because topical delivery of drugs to the eye is a much more viable process than it is to other parts of the body, because you can put fairly toxic things, atropine being an example. But phosphodiesterase inhibitors would be ideal drugs to test that concept, because they would elevate cyclic AMP and maintain it high. Another part to this that just confounds this, as you probably know, diabetics develop cataract. And they develop cataract in part because of changes in the osmolarity within the eye because of blood glucose. And that can be somewhat controlled, but again, it eventually leads to pathological damage. And another issue that we're just beginning to look at, and this leads to the second question, cataract surgery, which is common in this country, but not common in the third world, can also lead to secondary cataracts. So there's some, and that's probably the immune response. The second part is the antibodies. So we are developing phospholipid antibodies to the serine 235 because we think that monitoring that is going to be the key The issue in terms of human samples is most of the samples that you can get from cataract surgery is so damaged that it's difficult to do enough analysis to have, you know, a longitudinal study that can really tell you the benefit of that. But those are all things we're thinking about.
There is now an animal model. There's an animal model, a new one of neuromyelitis optica. I wonder if that's helpful in any way.
No, that is very useful. And also the mice that lack this ACAP that we talk about, ACAP2, which I left the name out because it would just confuse people, they have a visual phenotype, which is a combination of cataract and I forget, it's a retinal problem as well. I mean, it's in other places too, but it all fits.
Thank you.
Is that me? I did.
Very nice talk. I have a question. Is the interaction between the aquaporin channel and the ACAP transient or permanent? And if transient, what regulates the interaction?
Right. To the best of our knowledge, it's permanent.
Why do you need a permanent interaction? I mean, nature would just mutate the residue and make some sort of a constitutive form of phosphorylation.
But that's what we were just talking about. We don't think that happens so easily because the idea that you have the reversible phosphorylation to increase the peristalsis from one cell to the next is probably the explanation. In terms of the protein-protein interaction, we've calculated the KD as being in the order of, I think it's about 100 nanomolar. There's little or no off rate. So the KD for that protein-protein interaction is slightly higher than the KD of the ACAP for PKA. But we've measured that.
So, John, are you suggesting that the PK phosphorylation, is phosphorylating the target still bound to the regulatory subunit, or it just kind of flickers on and off quickly to do the phosphorylation? I mean,
don't know yet.
My understanding is that the regulatory subunit kind of puts an inhibitory peptide in the.
So it puts an inhibitory peptide into the
right.
active site, but the best substrate, bar none, is R2 itself. And R1 does not have, it has an auto inhibitor residue in alanine. So the way that we're about to address this, because this is pretty dynamic stuff, I mean some of it's going on, hopefully as we speak, is single molecule threat analysis by a completely different way to look at that, because it really, that's the next question. But there's definitely movement of that part, and I've been talking a lot to Susan about this, but that is the issue. And in fact, as far back as 1975, there were two back-to-back papers, one from Maura Rosen's lab, one from Ed Krebs's lab, that said exactly the opposite about that phosphorylation site. One said phosphorylation increased the protein-protein interaction, the other said it decreased it. So money's on what's going to happen there. But that release would be all it would take. We can certainly see, we don't see a gross conformational change, but there could be a subtle change. I mean, cyclic AMP is doing something, there's no question about that. I mean, there's all the beautiful work that's been done on catabolite activator to look at how it changes it in Susan's work. But you've hit the nail on the head. We think that's where the action is. Also, work that we have done, if you mutate the two arginines upstream of that, you get a constitutively active kinase. But the issue is it's not quite such a stable complex. But that's biochemically, because all of these things have been characterized so far by passing them over gel filtration columns. It's a pretty harsh approach.
So in all of the work that your lab has done on ACAPs, is there any evidence of peptides in particular that might break or successfully compete the R2 ACAP interaction?
Oh, that's how we started with HT31 peptides. And they have a 4 nanomolar to 850 picomolar binding affinity that can be used inside cells to uncouple, but they globally uncouple PK anchoring. And those have been used to define anchored PK phosphorylation events. but that could be just displacing the holoenzyme from the complex. One of the other things we've done with those peptides is using our kinase activity reporter, which is, there's so many reporters in there, it's hard to remember which one. If we displace with that, we decrease the maximum signal that we get, which would suggest that moving the complex away from the substrate gives you some effect. But you could be also moving something back in.
Okay. And then what about R1? What is the prevailing view on whether R1 is anchored or not? And do you think it's the same? Do you think it's different?
So R1 can bind to any ACAP. The off rate's slightly higher. The other thing with R1, it has an alanine in the active site, and it's 7-fold more sensitive to cyclic AMP. So we've done some measurements with R1. They're not quite ready for prime time yet, but it would appear that we can form the same complexes, but the sensitivity to phosphodiesterase inhibitors is slightly greater. So there could be more soluble C subunit with R1. But again, it depends on the amount in the cell, because R2 is the predominant form in any cell type. And if you overexpress I forget which way it goes, if you overexpress R2, you get rid of R1. Dr. H.
Here. Is it known if the unstructured linkers get modified?
Great question. They can be phosphorylated. The only issue is that from one species to the other, the phosphorylation sites in the kinases that phosphorylate them change. We've considered playing games like putting a well-defined protein interaction motif in there, but we've not done it. I mean, my idea would be to something that would either scrunch it up or stretch out, but we've just not done it, because it's beginning to get away from physiology.
So John, what fraction of pKa complexes are not bound to an ACAP in the cell?
So this is a hand waver. I'll tell you the experiment, but I think it's a pretty good one. So when we were doing, I can talk to, Tony's known this for a while, so when we were doing the solid phase overlays with fluorescent R2, we could do in situ overlays that we could use HT31 peptides to displace really well. When we did those experiments, we could show a huge number of sites, more so than the native sites. And also remember, MAP2 was the first ACAP identified, and there's a ton of MAP2 in every cell type. Plus, ACAP95, which is in the nucleus, although there's no R2 there, can sop up a lot of sites. So theoretically, there are more anchored sites available than ACAP PK complexes, which would fit with what we're talking about, I think.
So I guess, how then does free C subunit get into the nucleus?
I was hoping you wouldn't ask me that. I talked to Jerry about that earlier. I don't think it does under normal conditions, because two things to answer that. So I remember having many conversations with Dick Goodman, who was one of the people that discovered mammalian Krebs, and they always said that in order to get those experiments to work, they had to co-transfect a plasmid that overexpressed the C subunit. That was the only way they could get the experiments to work. And Jerry and I were talking about this earlier. I humbly would suggest that CREB could be called KREB, because it could be other kinases that do it. CAM kinases, RISC, MAP kinases. I mean, it's hitting serine 133, so it's possible.